We're going to talk about gluconeogenesis. We just kind of introduced it a little bit on last Thursday. Then we're going to get into pyruvate dehydrogenase. We're going to get into the mitochondria. Pyruvate dehydrogenase, the citric acid cycle. We're going to complete the oxidation of glucose. Take that glucose, the 6 carbons, convert all of them to CO2. Let's see, through glycolysis, have we converted any carbons to CO2 yet?
I don't think so.
No, we just made pyruvate, made a couple molecules of pyruvate. We did do a little bit of oxidation, right? But we did make a couple of NADH, those high energy electron carriers. We didn't actually make any CO2 yet. Okay, the reminders, the homeworks are due plunk, 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 plunk. This is kind of a busy week. No holidays this week, no planetarium shows this week, so it's busy. But then next week's our last week, right? Is that it? Man, it happens fast. Okay, midterm 4 on Thursday. Oh, one of the things I want to talk about, the other thing that is due, I think I have it set Wednesday night, okay, and it's basically giving you points for studying for the test, but it's a little bit more than that, is to make a pathway map assignment, okay? First of all, what is a pathway map? And I'm going to show you, there's actually a link on Canvas, but the link is kind of broken because Roche, the pharmaceutical company, has traditionally made these metabolic pathway maps where you take all the chemical reactions of a cell and map it out on a single great big piece of paper. But they're updating them, and they said, so they're currently not available. But I did find an old one online here. Just a sec. Here we go. Here's the map. This is in my little background slide for the course, right? So here are all the like this. Well, you can't see this. Actually, citric acid cycles, I think, is right about here. Let's zoom in a little bit. I think we can zoom in. Starting to see a little bit more. Where we go? Let's see. There's some pyruvate there. We might be getting close to somewhere we recognize. See if I can find glycolysis on here. Well, here's the citric acid cycle. Here. I can find glycolysis around here since that's the one we just did. Anyway, well, I'm not going to spend too much time on looking around for it on here. You can find that kind of thing. But here's the point. The point is, I wonder if I can not... I'm zoomed in as much. What a metabolic pathway map is you have a couple of metabolites, the intermediate pathway, and you link them together, showing what the name of the enzyme is between them, and any cofactors and that kind of stuff. And you draw the structures out on that. So for a metabolic pathway for glycolysis would have, well, we've got 10 reactions. Is it 10 reactions, 11 different metabolites, something like that, by the time you start with glucose and pyruvate? And you just link them all together, and you write the enzyme name, and then if they're making ATP or consuming ATP or making NADH or making ADP, you link them all together. So what I ask you to do for this assignment is to make your own metabolic pathway map, and then you're going to turn it in. And so what you want to do is hand draw it, okay? That includes glycolysis, gluconeogenesis, right? And so for gluconeogenesis, what we're going to see is that there's actually only 3 enzymes that are unique to gluconeogenesis. So you can just draw the backwards arrows from your glycolysis map. The homolactic and alcoholic fermentation reactions that we had, and the pyruvate dehydrogenase and citric acid cycle, which we'll start to go over today. So what do your maps should have? Chemical structures. You'll see some maps out there if you surf around on maps that just have the names. But this is a biochemistry class, so we like to think about the chemical structure and understand the chemistry of what's going on. And so draw all the chemical structures in there of all the reactants and the products. You don't have to draw the structures of ADP and NAD and all of those, but for all those intermediates in the pathway, you want to put the chemical structure. The name of the enzyme, so you have the structure glucose 6-phosphate, the arrow hexokinase, little arrow ATP to ADP on there. And then we'll talk about regulation, but identify typical regulatory steps and common activators, inhibitors of the regulatory step. So what I'm trying to get you to do is get your own mental map, right? And so if you draw it out, you draw out those reactions of glycolysis and these other, then you kind of, that way you put it in paper is kind of your mental map for what's going on. If I say aldolase, oh yeah, that's that reaction that's kind of over there in my map kind of thing. So the idea is that you begin to, by making a physical map, you generate your own mental map. It's kind of like a number line for those who do a math. Now, I don't know if you do math like with a number line in your head. Like if you have 3 plus 4, you kind of see where it goes on the number line to get to 7. Well, same kind of thing. It's like that. So that's the idea. So that is due Wednesday night. The idea is to basically do it before you have the get it in before the test, and then you'll take a picture of it and then submit that as a file on Canvas. Any questions on this? So it's something you can kind of just like work on as we go through this week, go through these pathways. Yes.
Somebody was asking on, do the different maps need to be connected to each other?
Yes, connect them all together, because that's a part of the idea is that we're going to see how these different things interact, right? So you're going to have glycolysis, and you're going to end up with pyruvate. Then you're going to move the pyruvate into the mitochondria, and then you're going to start linking that into pyruvate dehydrogenase in the citric acid cycle. Yeah.
When you say hand draw, can we hand draw on a tablet, or does
Yeah, you can hand draw on a tablet, right? What I don't want you doing is copying and pasting a metabolic map from someplace in there, or the structures in there. I want you to draw the structures because I believe that by drawing the structures, you think about the chemistry a little bit about what's going on. Right. Let's see, other administrative stuff. About 80% through that exam 3. I should be able to get this back to you tomorrow easy enough. I thought I was shooting to get them back today, but I didn't make it. I got close. Anybody check out the track and field trials on TV? The Olympic trials, track and field trials. And the track and field Olympic trials were held, I think, a couple cycles here at Sac State campus. They're up at the University of Oregon right now in Eugene. They kind of stole them back from us. Nike, I think, had something to do with that. But they used to be here on campus. Maybe in the year 2000, I went in the year 2000. They were here on campus. When were you born? 2002. Yeah. I was afraid of that. Oh, making me like this. But you know what was that particular year was what was known about, or sort of notorious about that year in track and field was the doping that was going on in that year. And this is a little bit of the chemistry of that doping. So these are anabolic steroids. Anabolic steroids are things that basically promote muscle growth. And here's the natural anabolic steroid, testosterone here. And here's a couple of analogs that have been known for a long time to have androgenic activity. So here's testosterone. This is dihydrotestosterone. So you basically take this double bond and you hydrogenate. It's also known as androstanolone. It has the street name of andro, basically, and it's more androgenic than testosterone, so it has a stronger effect. Here's a couple of well-known artificial anabolic steroids that have been used or abused in both people and horses and that sort of thing. So here's another one. Well, here's the structure of testosterone again. Here's another one called gestrinone here. And if you notice, it has this alkyne up here. And this was a well-known anabolic steroid, and there are tests for it that you could basically identify that. And so if you were tested, the doping agency would be able to identify that you have used this. But somebody figured out if you take this triple bond here and you just hydrogenate that triple bond here, then you get rid of it. There were two advantages. One is it's exceptionally androgenic, right? So just a little bit is very good. And because it hadn't been known, the analytical test wouldn't pick it up, basically, because it has a different molecular weight than the other ones. These are usually mass spec-based tests and a different molecular weight. So here's this like super steroid that couldn't be detected. So in the year 2000, it was like rife through rife through the track and field community, and people had it, people didn't, and eventually a track coach sent a vial of it to the doping agency and said, you might want to start looking for this kind of thing, and then everything blew up. This is the one that Barry Bonds was alleged to have used, and that sort of thing. Anyway, it's a little biochemistry related side story, given that it's track and field time, trials time. It was hot. That's what I remember. It was hot in the stadium, the Hornet Stadium here, right in this, with a weekend like it was. Oh, it was hot. Maybe that's one of the reasons it ended up going to your feet too. It just ended up getting too hot. All right, we were talking about gluconeogenesis, right? We were talking about how, occasionally or often, you're not just breaking glucose down, but you have to remake it. That there are organisms that basically have little carbohydrates in their diet, including people. Some diets have very little carbohydrates in the diet, but you still have to maintain blood glucose, particularly because the brain relies on blood glucose. So what you need to be able to do is convert amino acids back into glucose. Well, the amino acids get converted into pyruvate, basically, and then the pyruvate can go backwards. Well, all those reactions, those 10 reactions of glycolysis, are reversible except for three. And it was the pyruvate, it was actually number 1, the hexokinase reaction. Number 2 was the phosphofructokinase reaction. And the third one was the pyruvate kinase reaction. So we have to figure out how we're going to get around those 3 reactions. So let's see. Go back to our outline. We had, let's see, I had number 3 was metabolic fates of pyruvate. So 4 is gluconeogenesis. Gluconeogenesis doesn't happen in the muscle. It's very tissue specific. It happens in the liver because the metabolic role of the liver is to help maintain blood glucose levels. Muscles, not so much. The muscle just wants to consume glucose, make ATP, be able to drive muscle contraction. Okay, so let's look at the enzymes. So we have to go backwards, right? So the first one we're going to talk about is to try to go from pyruvate to phosphoenolpyruvate. So this is the reverse of the final reaction of glycolysis. And again, this is one that under physiological conditions, you can't just, the pyruvate kinase just won't go backwards. You have to kind of go a different way. And it's actually a 2-step reaction. We first take our pyruvate, and we carboxylate to form oxaloacetate. This enzyme is known as pyruvate carboxylase. And it requires, energetically, it's going to require hydrolysis of an ATP. So I'm just going to put ATP goes to ADP plus PI. So this is coupled to the hydrolysis of ATP. So this usually happens in the mitochondria typically, and there is a whole system then for moving the oxaloacetate out from the mitochondria into the cytosol. We won't get into it, it's called the malate shuttle. We won't necessarily get into that for this class, but we basically, so this typically happens into the mitochondria, and the oxaloacetate moves indirectly via malate back out to the cytosol. Then, the second reaction decarboxylates again and generates our phosphoenolpyruvate. This also requires the hydrolysis of a nucleotide triphosphate. In this case, it's actually GTP. So one of the things you notice, so in the glycolytic direction, in glycolysis, the last reaction of glycolysis was the conversion of phosphoenolpyruvate to pyruvate, and that was associated with the synthesis of 1 ATP. Okay? Now that we're going backwards, in order to go backwards, it actually takes 2 ATP equivalents to remake that, right? So it's energetically expensive to go back. So one of the things you don't want to have happen is in the cell, you don't want to have the pyruvate kinase reaction, which is going this direction, making 1 ATP, going at the same time this resynthesis of phosphoenolpyruvate is making. Because if you just get a cycle going, you're just going to be going through ATP, right? You're going to generate 1 but burn 2, generate 1 but burn 2, generate 1 but burn 2, and then you're just going to keep using up ATP. So you need to have careful coordinate regulation of glycolysis and gluconeogenesis. So when glycolysis is on, only it's on. And when gluconeogenesis is on, only it is on. And we'll get into mechanisms of that in a little bit. And we're probably most focused on other steps, but I just wanted to bring up that concept is that you basically can't have things just cycling back around. Otherwise you just burn up ATP.
Did you want us to know an enzyme on the second step?
Yes, this is phosphoenolpyruvate carboxykinase. What a name. I'm going to put PEP. So this is PEP here. Carboxykinase. All right, so we're trying to remake glucose from pyruvate, right? And so we've redone basically the last reaction of glycolysis by remaking phosphoenolpyruvate. Now the next several steps, which if you go back and look at see if I can remember it backwards, it's always hard to remember. It would be like going to enolase, phosphoglyceromutase, then enolase, then phosphoglycerate kinase, glyceraldehyde 3-phosphate dehydrogenase. All of those are reversible, right? Those can just go backwards. So you can use the same enzyme to go forwards or backwards. The next irreversible step is when we have to, even the aldolase, the one that split it into the 2,3 molecules, that's reversible too. So the next irreversible step is fructose 2,6-bisphosphate, 1,6-bisphosphate to fructose 6-bisphosphate. That's the second thermodynamically irreversible step that we have to have a workaround. And so here is our fructose 1,6-bisphosphate. And to go backwards again towards our fructose 6-phosphate. We're just going to cleave that phosphate off by hydrolyzing it off. We're just going to hydrolyze this phosphoester right here. That's an energetically favorable reaction.
Professor, is that a negative or an equals or what's?
Oh, 2 minus.
Oh, okay, thanks.
Yeah, I'm giving you all sorts. I could have put a P with a circle there, or a PI, and now I'm giving you another one. And this enzyme is fructose 1,6-bisphosphate phosphatase.
I have a question. So you said that there were 3 non-reversible reactions. Are we adding these to the list?
Yeah, so that first one is that one. That one's this one. In glycolysis, you know, in glycolysis, we're taking phosphoenolpyruvate to pyruvate. That's the last reaction. In glycolysis, we're taking fructose 6-bisphosphate to fructose 1,6-bisphosphate in the enzyme known as phosphofructokinase. And so this is the reverse of that. Now, the forward reaction in the glycolysis direction is going to go from here to here. That requires an ATP. And again, we don't want recycling because we're going to use an ATP this way, but not get it back going backwards because we're just going to cleave it off. And if you just cycle back and forth, you're just going to cycle through ATP. So again, making that need for coordinate regulation of glycolysis and gluconeogenesis. The third step is what is the first step of glycolysis, but we're going backwards. So this is glucose 6-phosphate to glucose. we're going to do a very similar kind of reaction where we're just going to take water, hydrolyze off the phosphate, and remake glucose. And this enzyme name is glucose 6-phosphate phosphatase. Yeah.
Why is it PO42 minus?
Why is it PO42 minus? Well, so it's a little bit. Let's we can get into that.
It's not important, matter.
Well, it's so what we're going to get is this. The pKa's of this. Remember, the pKa's is like. It's such that one of them's definitely going to be protonated, one's going to be deprotonated, and the other one's going to be kind of half protonated. So it's really PO for 1.5 months. There isn't really an exact number. So this one, you know, at our pK, this one's going to be kind of on and off, and so it's going to have like a partial negative charge. All right, what I want to do, just because I kind of get, some people might be a little bit lost, which is understandable. That it's like, I do this all the time, right? Every semester I'm doing it like this, so I get it like this. It's easy for me just to whip off these names and then forget that when the first time you see it. It's like, you know, driving to your house in your neighborhood. If you know it really well, you can get there really quick, but if you've never been there, it's a little bit dark. So a little bit harder. So I just want to kind of give a really brief just overview of glycolysis and then showing what we're doing going backwards. So and maybe I won't draw all the structures. Glucose, so this is in the glycolysis direction. Glucose 6-phosphate. Remember, we use ATP to go to ADP. And then we have fructose 6-phosphate. Then we go to fructose 1,6-bisphosphate. This takes another ATP to ADP. Then we go to glyceraldehyde. Phosphate plus our dihydroxyacetone phosphate, and then that gets converted to the glyceraldehyde 3-phosphate. Then we get 1,3-bisphosphoglycerate. Then we have 3-phosphoglycerate. This actually gets us an ATP. Oh, this one too, we end up taking NAD plus, making NADH. Then 3-phosphoglycerate, then 2-phosphoglycerate, then phosphoenolpyruvate, then pyruvate, and then this is
ATP, ADP?
Yeah, this is
ADP.
ADP makes ATP. Yeah. All right, so those are the 10 reactions of glycolysis. So to go backwards, what we're saying is from that first one that I gave you, the pyruvate to phosphoenolpyruvate, that's that two-step reaction. Basically go here, and we go through oxaloacetate to go backwards. And this one takes ATP, ADP plus PI. This one takes GTP. And then CO2 comes on and then comes off. All right, so this is what I mean. So now we're going backwards here, right? And we have to bypass around because under the conditions of the cell, this is essentially the pyruvate kinase reaction here is irreversible. So we have to do this two-step workaround. But these guys are all reversible. We can go backwards here, 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 we can go backwards here.
Does it use the same enzyme to go back?
It uses the same enzyme. It uses the same enzyme.
Okay.
Exactly. Like this. And so we just by altering the ratio of substrates to products, we can move it, go backwards here. Now we get to this stuff. This is the one that is, this is the phosphofructokinase, known as PFK. This is also an irreversible reaction. So that was irreversible reaction 1, 2, this one like this. So here, to go backwards, we're bringing in water, and then we're yielding PI. But this one is reversible, we're just fine here. And this is the same thing, water, and we're yielding phosphate. Does that help a little bit? Let's see what we're doing going backwards.
And so, just so I have to clear, phosphogluconeogenesis, what is the whole word of that thing?
Gluconeogenesis.
Okay, no phosphate at the
Yeah.
beginning. It's just the process of it going backwards, and then glycolysis is it going forward?
Exactly, exactly. Neogenesis, making new glucose. Then if the liver cell, what you're going to do is you're going to take that, you're going to dump it back and dump it into the bloodstream. Yeah. Yeah.
Professor, were you saying that if people don't eat glucose, that they can make pyruvate from amino acids?
Exactly, exactly.
And then go backwards to get
Then
the
go
glucose?
backwards, yeah. And a little bit more detail. So amino acid metabolism will feed either into pyruvate, amino acids, or into oxaloacetate. So somebody with a low carb diet that has plenty of protein, their blood sugar is just fine. All right. Now, but here's the thing that I was kind of harping on, and we're going to focus it on this step, is that you can't have glycolysis and gluconeogenesis going on at the same time, because if you do that, you're just going to burn through ATP. If you had the phosphofructokinase reaction, this one going through, you're gonna make ADP, and then that phosphate on the way back is again. So the overall reaction, if you go through here, is just to take ADP plus ADP and make ADP goes to plus water goes to ADP and plus PI. You just burn through ATP. So you don't want both of these processes going on at the same time. You have to have coordinate regulation. And so we're going to focus on that. The coordinate regulation, particularly at the, and let me maybe draw this out. So this is phosphofructokinase. In the glycolytic direction, in the gluconeogenesis, this is fructose 1,6-bisphosphatase. So we're going to talk about how does a system make sure that when PFK is working, this one isn't, or when this is working, that one is. At least one mechanism here. All right. One of the mechanisms, there are multiple mechanisms for this, but the one that's probably the most cool and the one that's cited is allosteric regulation. So I'm going to make this another little what we're at. Gluconeogenesis 4, so this is 5. This is coordinate regulation. Of glycolysis and gluconeogenesis. And our main focus is going to be on the phosphofructokinase, PFK, and then the fructose 1,6-bisphosphatase. How do we make sure that PFK, when PFK is on, the bisphosphatase is off and vice versa? And we'll start by talking about allosteric regulation. Remember, allostery, that's when you have some other molecule that's going to come in and move it from the taut to the relaxed form, make something active. Allosteric regulation by another molecule that's a fructose structure, okay? It's actually fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate. What the heck is that? So now with fructose 2,6-bisphosphate, remember this is carbon number 1, 2. This has an O here. Now we've got our phosphate is going to be at the number 2 position rather than it is in the number 1 position when we're doing the glycolysis. So this molecule is no longer it's not an intermediate in the pathway, but rather is a regulatory molecule. So this is fructose 2,6-bisphosphate. And so what it does is it activates phosphofructokinase. And that's going to ultimately promote glycolysis. And at the same time, it's going to inhibit the fructose 1,6-bisphosphatase. And that inhibits gluconeogenesis. Okay, so this is basically a nice molecule that acts as a switch. When it's around, it's going to activate phosphofructokinase, promoting glycolysis, and at the same time shutting off phosphofructokinase. When it's not around, then the phosphofructokinase is unstimulated, basically isn't very active, but this one is uninhibited, and so it's going. So when you don't have the fructose 2,6-bisphosphate around, then you're going to promote gluconeogenesis.
So, of course, these things can't run in tandem with each other at the same time, but can they both be off at the same time? You know, assuming the person's like still alive or whatever.
Oh yeah. Can they both be off? Okay, so yes, and this also kind of recognizes, and we didn't when we were talking about enzymes earlier, we didn't get to this, is that there are multiple layers of enzyme regulation. So there's this sort of allosteric regulation that we might have, but there can also be just the amount of enzyme that's around. So that might be under transcriptional control. There can be things about how much are they, what's the degradation rate of these enzymes, that you can change the amount of that enzyme either by promoting its synthesis or promoting its degradation. And so there are other ways of regulating these enzymes that might shut them both off. For example, if you shut down transcription, and so that they're actually, neither one of them weren't there. Or more likely, they might actually both get turned on. An example of that is in cancer biology, cancerous cells have very high glycolytic rates. They go like crazy. So what you find is there's actually a transcription factor that is activated in cancer cells that promotes the transcription of all these glycolytic enzymes, so that there's a high rate of glycolytic enzyme. There's high rates of glycolysis both because those rapidly dividing cells need a lot of energy, but they also need, the other role of glycolysis isn't just to generate ATP, but all those intermediates also serve as building blocks for building the amino acids and other components in the cell. So, so transcriptional control can be very important in glycolysis as well. But that's usually on maybe a day time scale, while this kind of thing is on a minute time scale. This is the kind of thing when you're more on the minute time scale. Okay. So, but we kind of kick the can down the road a little bit and saying, oh, well, there's this molecule that basically will turn one on and shut the other one off, right? Then the question is, well, what regulates how much of this molecule is around here, all right? And so there is another enzyme that is known as phosphofructokinase 2. So fructose 2,6-bisphosphate levels are controlled by phosphofructokinase 2 and fructose 2,6-bisphosphatase. So there are two other enzymes. So we're going to take our fructose 6-phosphate here. Here is our fructose 6-phosphate. And there is another fructokinase. Our fructokinase that we use in glycolysis, that's going to phosphorylate this position. Well, there's another one that's going to phosphorylate that position. So it's going to take ADP, just like the other phosphorylate, except instead of putting it at the 1 position, it's going to put it at the 2 position. This enzyme is, again, phosphofructokinase. I'm just going to write PFK2, phosphofructokinase 2. And guess what? There's another enzyme activity that will degrade the phosphofructokinase by hydrolyzing it, just like before in this. So we're going to get PI out here off. And so this is fructose 2,6-bis phosphate phosphatase.
So, Professor,
Mm-hmm.
if those two enzymes are working against each other, is there something that controls how much of those there are?
Exactly, exactly. So we're just kicking the can down the road some more, right? We're basically saying, so what? So this is what's really cool about this, is I'd like to take some time about this. Is that actually, both these enzyme activities are on the same polypeptide, on the same protein. But this is kind of this protein is like Dr. Jekyll and Mr. Hyde, right? Sometimes it acts as a phosphofructokinase, sometimes it's like this. What controls whether it's Dr. Jekyll or Mr. Hyde? What controls it is its phosphorylation state. So, so it is actually this PFK2 F26 bisphosphatase is a bifunctional enzyme. When it's phosphorylated, it acts as the fructose 2,6-bisphosphate. Okay, so let's see what happens when that protein is phosphorylated. When that protein is phosphorylated, it acts as a fructose 2,6-bisphosphatase. So that's going to take this, and it is going to hydrolyze it to this, so we get rid of our fructose 2,6-bisphosphate, right? So if we don't have the fructose 2,6-bisphosphate, we're not going to activate phosphofructokinase, and we're not going to inhibit. So the net reaction is to promote gluconeogenesis, all right? So all of this leads to the ultimate thing is promoting gluconeogenesis. And the opposite is when it's not phosphorylated, it acts as a PFK2, a phosphofructokinase. So how does that work? Well, we got PFK2 here. That's going to make more of this fructose 2,6-bisphosphate. When we have more of the fructose 2,6-bisphosphate, we're going to activating the phosphofructokinase, inhibiting this phosphate. We're going to promote glycolysis. So this is going to promote glycolysis.
Professor.
Yeah.
So for this enzyme, are there two enzyme domains, or is it just one in phosphorylation?
So actually what it is, I didn't draw a picture, is it kind of looks like this. There's like a link and there's like a link here. And so there's a PFK2 domain and then a FBPase domain like this. And then I can't remember exactly where the phosphorylation site is to actually turn one to turn it off. Okay. So Danielle, what's the next question? How do we know when it's we know what's going to control the phosphorylation state? So now we can actually maybe get something that's a little bit more organismal, that's bigger. Still a multi-step process. We're not going to go through all the steps. But basically, glucagon ends up promoting the phosphorylation. Glucagon is a hormone that is made by the pancreas when blood sugar is low. Low blood sugar stimulates glucagon, phosphorylating this, then promoting gluconeogenesis. So if you think of that, it kind of makes sense. Blood sugar is low, in context of the liver, blood sugar is low, it's going to promote gluconeogenesis. That's going to result in the liver dumping more glucose back into the blood. Okay, under high blood sugar, then we have insulin, which is going to promote the dephosphorylation of that. And we're going to have the other, we're going to actually promote glycolysis. Okay, in between, there are whole courses basically talking about this process here. So I don't want to act like that's like a real simple, straightforward process. There's a lot of variability of what happens in different cell types and other players in the whole thing. And for those of you who had a cell biology process, it's via that whole cyclic AMP-mediated phosphorylation cascade, and you go through that. But for this course, we don't get into that detail. And it really is kind of, that detail is a course in itself.
Professor?
Yes.
They've started making, I don't know how widely used they are, but they've started making insulin pumps that are pumping two different things. I wonder if they're pumping the glucagon as well.
Oh, that's interesting. Yeah, I don't know. I don't know. Yeah, so that you basically have an artificial pancreas there that's redefined.
Well, yeah.
Okay, so yeah, let's see. I think at this point where we'll start with this is, okay, so we talked about coordinate regulation, and I wanted to focus, because this is kind of a neat story, right, with a bifunctional enzyme, this other regulatory molecule, and all that kind of stuff. But I don't want to lead you with the idea that's the only other way of regulation. And so I'll just to be sort of straightforward about it and honest without getting in the details, just mention that there's also transcriptional control, other allosteric regulators, and those sorts of things are going to be dependent upon the particular cell type. You know, so for example, you might have pyruvate feedback inhibiting the PFK in certain cell types, and that sort of thing. And then the other thing I'm going to do, just to keep from getting in too much detail here, is just to also say that while you don't have this fructose 2,6-bisphosphate, you also have, you know, transcriptional and allosteric regulation of those other two steps of the hexokinase and pyruvate kinase steps. Actually, there's a whole interesting story around pyruvate kinase. Humans, pyruvate kinase, there are 4 different isozymes, and some are expressed in the liver, some are in the muscle, some are in others. So actually, there's this interesting story about this, again, related to cancer metabolism. There's an isozyme of pyruvate kinase that is expressed during embryogenesis, right? During early stages of human development where there's rapid cell division. Again, very high glycolytic rates, and there is one particular isozyme of pyruvate kinase that has high activity levels, basically, and it's not allosterically downregulated by a lot of molecules. So it's like a super pyruvate kinase. And then its expression in normal human development, its expression goes away. But in some cancerous tissues, again, that have high glycolytic rate, that particular isozyme will be reactivated, and that'll be the one expressing. So there's been a lot of work trying to address that isozyme and see, is there a way you could selectively inhibit that isozyme to try to inhibit glycolysis in cancer cells? All right, we'll stop there, we'll take a break, and we'll kind of leave our glycolysis gluconeogenesis story here. And then when we come back, we're going to jump into the mitochondrion, and we're going to get into oxidative metabolism. All right, so let's take a, I don't know, 5-minute break here.